I wanted to start today with some discussion on the foundations of basic genetics. I told you guys that we would be getting some more distinct clarity on some of these vocabulary terms, so I wanted to make sure to give this to you guys, but hopefully a lot of this is review. So one thing that we need to make sure we understand is exactly what a gene is. And so in looking at a gene, please recall, let me get this, there we go. Please recall that a gene is located in a locus of a chromosome. So that's the location of the gene. So maybe you find one gene here, and the second gene is very close. But remember that genes are made up of specific sequences of nucleic acids or nucleotides. So they're made different by their A's, C's, T's, and G's. Okay, so we have our DNA that codes for our mRNA, and then our mRNA, remember our central dogma, is read by the ribosome, and that is where we get our protein. Remember that the order of mRNA, right? It's read in groups of 3, and that's how we figure out what amino acid is brought out. And so if we have certain amino acids used to make our protein, then the order of that is very important so that we can have the protein have its correct structure. We've used this term allele quite a bit, but remember that this is just the alternative version of the same gene. So we're looking here at things like flower, and so they occupy the same locus on homologous chromosomes, right? So they're in the same spot, but they're going to have a slightly different sequence of DNA, right? So if we're looking at our flower color here, here's our red flower, our pink flower, and our white flower, and they all have the same, they all have a gene for flower color, right? But the allele is going to be what is presented as a result of their gene. So maybe in their DNA they have ATTCCCGAGTACGGT, but this one little difference here gives them the allele for the dominant form of the gene, or red, which will create a different set of proteins than you would get if you had these 2 T's in place of those C's, which might give you that recessive form of the gene. Form of the gene, or recessive allele, that makes a different protein that gives you a different flower color. Okay? Similar in eye color, right? We could say someone has the gene for eye color, but they have a few slightly different base pairs in their DNA, and that DNA gives them a brown eye color over a blue eye color. So here's just kind of another version of that. Here's the flower color gene. And we have the DNA sequence within it that has this specific marking. Make sure you keep your eyes on those. We go through transcription, translation, and protein folding, and then there is ultimately a protein that is made as a result. That protein makes pigments, and because of this one nucleotide base change, we get purple flowers. Whereas if we have the allele for white flowers, we still have the flower color gene, but we have a DNA sequence that has a different nucleotide in here. Goes through still transcription, translation, protein folding, etc. And we get a protein that makes pigments, but instead of actually making this purple pigment, we actually have a protein that doesn't work very well. And so it has a loss of function because its shape is quote unquote wrong. That's what we're going to find oftentimes, is that recessive genes are typically a result of just an improperly working protein. And so that's where we come into talking about dominant and recessive. So a dominant allele is a variation of a gene that will produce a certain phenotype even if the individual only has one copy. We always write this as a capital letter, okay? So if here's grandma, grandpa, grandma is homozygous dominant, grandpa is homozygous recessive, their kids will still have that capital R, or the allele for brown hair from grandma, and so they're still going to express that brown hair allele. A dominant allele is typically for a functioning protein, okay? The example of this is that darker hair is dominant over blonde or red hair, because having even one copy of the dark hair allele will code for enough protein to make the hair look dark. Again, if you have two copies of the recessive gene, the recessive allele, then you're going to have maybe a protein that doesn't work quite as well. And if you get two copies of those, then you're not going to get the colored protein in there. Please note, I did put on here, this is a simplified example. In reality, hair color is polygenic. Remember, polygenic meaning that it is coded for by multiple genes. In fact, most of our traits are polygenic. A recessive allele is going to be something that is it will show up whenever you are homozygous for that phenotype. So the likely cause is that it is for the allele codes for a non-functioning protein. There's actually going to be two causes. Don't write all of the detail in the examples, okay? The example is going to be a protein called MC1R. So this is our skin color protein, right? This is our melanin protein that we've looked at before. This also helps to code for hair color. The dominant allele codes for a functional protein. As long as there is one working MC1R gene, the hair won't be red. The recessive allele codes for a non-functioning MC1R protein. That means that it's just not making the hair color. If both copies of this gene are the recessive allele, then no functional protein is made and no red pigment is removed, so the hair can be red. Another example of recessive allele is that it's a variation of a gene that must be homozygous when inherited in order to be expressed. So the recessive allele is the normal one and the dominant allele is a broken version. And again, I like to call this more, because what is normal, right? But if I could put this here, I'd also call this wild type, okay? So what we see in the majority of the population. But we need two copies to get the job done. So an example is achondroplasia or dwarfism. This is a broken version of the FGFR3 gene. The normal job of the FGFR3 gene is to code for a protein that prevents bone growth. And the broken version of FGFR3 is hyperactive. It tells bones to stop growing even when they should be growing. The hyperactive FGF3 protein causes a person's bones to be much shorter than normal. Even if a person has a normal copy of FGFR3, the broken version sends a signal that's just too wrong, which makes A dominant. So this is actually kind of an example of how the reverse can be interestingly, how the reverse, like the normal coding protein is actually the recessive one. So if you have even one allele that's dominant here, it will get the different version of the trait. Please note some common misconceptions, right? Recessive alleles are less frequent in the population, and also only recessive alleles cause disease. Nope, get that out of your head. For right now, you'll notice there's a section on codominant alleles. Please skip that section. Please remember, humans are diploid. That's 2N, which means that we have two copies of each chromosome and therefore two copies of each gene. So on chromosome number 1, right, we have hundreds of genes, but let's say that there's a gene A that you got from your mom and maybe a allele A that you got from your dad. So here's that example, FGFR3. So this is on chromosome 4. So here's the locus of the FGFR3 gene, okay? Since there are two chromosome 4s, there are two copies of this gene. So if you receive even just one copy of that dominant allele, then you will present with dwarfism. If you receive two, this actually results in death. Here's the locus for the MC1R gene. That's a typo on my part. So the locus for that one is here. And since there are two chromosome 16s, there are two copies of this gene. Here are some of those words we were using in class. Heterozygous is when you have two different alleles. Remember that term, prefix hetero, meaning different. So that could be big B, little b. We typically try and write the capitalized letter first, okay? But let's say that, on your locus here for the B gene, you have a dominant one, and on this locus you have a recessive one. The individual's mom and dad passed on different version of the gene. So this is notated by one capital letter for the dominant allele and one lowercase. Now homozygous, this is when you have two of the same alleles of a particular gene. So this could be homozygous recessive, right? Or homozygous dominant. So if you received a dominant version from both of your parents, then you would have the genotype of capital B, capital B. I just want to emphasize here, we have two more slides, but true breeding is genetics language. That means the parents always produce offspring that have the same phenotype. This means that the parents are homozygous for every trait, okay? So if you had two parents, they would have offspring that were all homozygous dominant, okay? And then finally, the phenotype is the observable characteristics of an individual resulting from the interaction of its genotype with the environment. That's important. Remember how genes can interplay with the environment there? It's that epigenetics piece. So these are all different things that can play a role in how genes are expressed. But notice how I'm putting here, okay, here's our genetic makeup. These are the alleles, right? So their genotype is that they are homozygous dominant. And then this fly gets the phenotype or the physical trait for no wings. Over here, here's an example of homozygous recessive. They're going to be able to get wrinkled wings. And then notice our heterozygote. They each have a large W present, so they still code for the phenotype of normal wings. However, let's say that these two had a baby, they'd still have a chance of having a homozygous recessive offspring. In fact, it'll be 25%. We'll go over why that is when we address Punnett squares.